Good morning, one and all. Myself, Mrs. Smita Sharke, Assistant Professor from Department of Pharmaceutical Chemistry, and glad to introduce our prestigious alumni, Mr. Sourav Suryawanshi. He is a second speaker of our today's one-day national level webinar. He did his undergraduate graduation from Sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research, Nasik. Currently, he is pursuing his M.Tech in Pharmaceutical Chemistry and Technology at ICT Mumbai at the Bhubaneswar campus. He is having different instrumentation skills, which include some modern analytical techniques like FTIR, ATR, HPLC, DSC, particle size analyzer, zeta potential analyzer. He also expert in some of the advanced techniques like XRD, SCXRD, lyophilizer, and texture analyzer, and so many. His academic achievements and awards include he got Student of the Year award in the undergraduate and also qualified ICT entrance and NMI-MS entrance. He also stood first in poster presentation in World Pharmacist Day in 2024. In academic contribution, he attended many conferences including BRICS Youth Council Pre-Consultation KIIT in 2025 and attended International Conference on Materials for Circular Economy in 2024. Today, Mr. Sourav Suryawanshi is going to talk on a topic of nanovesicular drug delivery systems pioneering the future of precision therapeutics. With this, now I request Mr. Suryawanshi to start the presentation.
Yes, first of all, I would like to say
Yes.
First of all, I would like to say thank you so much, Professor Sherke ma'am, for the generous introduction. It is an absolute privilege to return back to my alma mater as an resource person for this prestigious one-day national webinar on the integrative therapeutic innovations, VND pathway, nanovesicular system, and drug discovery, organized by the Department of Pharmaceutics under the aegis of IQAC at the rate MSG Copper. I would also like to thank Mr. Bambre sir, the coordinator, and the IQAC coordinator, Dr. Vice Principal Dr. Pingre sir, and our favorite and my favorite Honorable Dr. Principal Amrutkar sir. Now, I'm really happy to share my thoughts and share some my research work or my knowledge about the nanovesicular drug delivery system, the pioneering the future of precision therapeutics. Now I'm going to share my screen. Is my screen visible to everyone?
Yes,
Am I
screen
audible
is
to everyone?
yes, you are audible.
Yes.
Your
I'll make it.
screen is also
I'll make it
visible.
full. Yes, ma'am.
Yeah.
Yes.
You are
Good
visible.
morning, one and all. Good morning, respected present people here only for the one-day national webinar. So today I'm going to focus on the topic that is very informative and that is very knowledgeable topic for the Sutics point of view and the pharma point of view. That is the nanovesicular drug delivery system that is pioneering the future of precision and therapeutics. Now, moving to the next slide, that what should we learn today from this topic of the presentation. Here are some learning objectives. So by the talk, you all will be able to name the major vesicle types, then there is distinguished feature, understand the main formulation and analytical techniques to develop them. Then you also understand the types, as well as learn some formulation and characterization techniques, explore targeting and stimuli responsive strategies, and also we can get some knowledge about the review clinical market example and regulatory challenges. Now, the introduction part. Now, the major question: why nanovesicles? Why the nanovesicles? As we all know, the nanovesicles are basically they are ranges in the nanometer, and they are look like cargo ships. As we know, cargo ships transport one material from one country to another country. That's like these nanovesicles are like nano ships, or we can call it as cargo ships, that are completely biocompatible with our cell membrane, as well as they protect the therapeutics which are encapsulated inside it, and they will transport them to the site whichever wherever they are need. Okay, so the nanovesicle, basically why we introduce these nanovesicles in the pharma field. Basically, nanovesicles are helpful. They protect the drug or the biologics from the degradation and ensuring its precision and targeted delivery to the disease sites. Okay, so as we know, the traditional therapeutics or formulations are often failed to drug the deliver at the specific site or targeted site. So in this manner, by formulating the nanovesicles, we can advancedly target its delivery, and we also can design it according to the control release. And in this manner, we can enhance the safety and the efficacy by targeting the specific physiological site, as well as we can optimize the pharmacokinetic profile of the body through the controlled and sustained release by the diffusion and the degradation. Now we can call it as a versatile carrier system. As the nanovesicle, as we know, the nanovesicle can carry both hydrophilic as well as a lipophilic moieties, and significantly it will also increases or enhances the bioavailability of orally soluble compounds, while it also reduces its systemic as well as some hepatotoxicity of the drug molecules. Okay, so it will also expand the therapeutic potential, and they enable the effective delivery of the complex molecule. For example, there may be some peptide molecule, there may be antibiotics and so on. Okay, so treat and it will reach the intracellular infection and target and the and it can also help to target the specific tissue, specific organ, specific cell, and specific disease site. Okay, so now the next part, that is the what is the importance of this vesicular drug delivery system, and why the recently the scientists and whole the pharma industries are focusing on the vesicular drug delivery system in pioneering the therapeutics. There are some basic important points regarding it. The enhanced efficacy. Okay, so the drug will directed toward the specific cell, and it will also increase the effectiveness and the precision of treatment at that particular site, as well as it will reduce the side effect. As we know that there are some the conventional route of administration, such as oral route. So in oral route, we all have studied that the fast metabolism, so drug degradation, as well as some GIT related disorder may occur due to this traditional route of administration. But due to the vesicular drug delivery system, what we can suppose it has a beneficial over that, it can minimizes the exposure of healthy tissue to get to the drugs. So the drug can only target and only treat the infected tissue only, not the healthy tissue. So it will also lower the adverse reaction. And the main important point about the vesicular drug delivery system, that it will improve the patient compliance, it will reduces the side effect and higher, and it will stick to the adhere to the higher patient safety and efficacy. Now we can call it as a cost effective solution as well. And because it target this targeted therapies will reduce all the healthcare costs associated with the treatments and the diagnosis, all related topics. So, and we can call the important point about the vesicular drug delivery system, that is the optimized drug dosage. Okay, so as we know, in traditional drug delivery system, the drug doses are maximum, the concentration of drug are maximum in the dosage form, as well as the dosing frequencies also increase at that time. But by formulating that particular drug in a particular vesicular drug delivery system, we can also minimize the dosing frequency, also minimize the dosage concentration. Okay, the versatile application, as well as we can see it can be applicable in various field of a healthcare system, that is neurology, oncology, cardiology, and various infectious diseases. The advanced research that promote the innovation and development of the new drug delivery and technologies. Okay, now the historical evolution of a vesicular system, as we all know, the it has some historical background. Now that the vesicular system evolved from 1960s, nowadays we can see many various exceptional work in the vesicular system. But now we have to look back toward the historical evolution. In 1960s and 1970s, the era of discovery, where the Bangam and its lab mate or it and its that co-worker, they first of all observed the swollen phospholipid, and they called it, and they named it as a liposome. In liposome, it was coined, this term was coined by Mr. Sesa and Weissman in 1968. Then in 1971, they establishes, or they propose this particular vesicle, this phospholipid vesicle, swollen phospholipid vesicle, as the liposome, okay, for the drug delivery system. And in 1975, after this breakthrough of liposome, in 1975, the niosome, that is a novel vesicular system that has distinguished features than the existing liposome. So niosome by the L'Oreal in 1975. Then the era of optimization and translation has come in 1980s and 1990s. In 1980s, Stealth Tech, he developed the PEG-lated. PEG is a type of a polymer, that is polyethylene glycol. Okay, liposome to evade the immune system and prolong the circulation. So they do some research work in the existing liposomal system, and they found that they can prolong the circulation period of this vesicular system in our systemic circulation. So the drug will release sustainedly, and will it remain in the blood circulation for the time. And in 1992, the Civic and his co-workers, they invent a transfer zone, that is ultra deformable and elastic vesicle than the liposome. Okay, it's basically the first they propose the liposome, and after that the scientists and the researchers have worked on the liposome structure. They made some changes, made some transition, made some various add some various excipients, and they evolved this vesicular system to another vesicular system. In this way, in 1992, they introduced the transfer zones for the transdermal delivery. And in 1995, the first marketed preparation, that is Doxil, the first clinical liposomal nanomedicine, were introduced in the market by the market, and it get approved by the US FDA. And in 1996, the Tau 2, they developed the ethosomes. Okay, basically it's a evolved form of liposome, just like other, that is transfersome, transfersome, niosome, just like that the ethosome. And ethosome has a special characteristic that it can penetrate deep inside the skin and deliver the drug across the dermis layer, and that drug will enter into the systemic circulation. Okay, so the next part, that is the again the next part of the historical milestone, that the era of advanced carriers, then 2000 to now present date. Okay, so synthetic evolution where occur the polymerazomes, that is a block of copolymer, means instead of they using phospholipid as a bilayer for the vesicular system, some scientists done some experiment. They done some variation for the composition of the vesicular system. They use various polymers, and that block copolymer with a high stability and the complex structure, they form the polymerazomes. And the utilization, with the utilization of this, they come with a new idea, that is new novel idea of cubosome as well as hexosomes for the poor soluble drugs to reach to the specific site of action. Okay, so there is one term, that is theranostic. Nowadays we can see that theranostic means the therapy plus diagnosis. So this vesicular system doing an exceptional work in the field of pharma with the help of this new technique, that is theranostic, that is emergence of smart vesicle, that combines diagnosis as well as therapy. It will diagnose the particular disease as well as deliver the drug at that particular site, at the particular cell and specific tissue. Now, the next part, that is a taxonomy of nanovesicular system. As of now, we know that nanovesicular systems are self-assembled lipid-coated nanocolloid, natural as well as synthetic cargo ship-like structures. Okay, that they are similar to the cells. Okay, and that encapsulate the various pharmaceutical, cosmetic, and food industry products. So the dynamic and architecture allow to adaptable to topical as well as transdermal administration, and it will also facilitate the personalized drug delivery system in the new era of therapeutics. Okay, so basically the nanovesicular systems are classified into two categories, that is lipoidal and non-lipoidal. From the name we can get knowledge, we can get some knowledge about that. The lipoidal means the, in that the building blocks, the bilayer are made up of phospholipids in the lipoidal nanovesicles, and in non-lipoidal nanovesicle, the shell, that is the lipid bilayer, that is our hydrophobic bilayer, in which the aqueous core, that is hydrophilic aqueous core, is present. That lipid bilayer is made up of non-ionic surfactant as well as some polymer, and we can call it as a synthetic amphiphiles. Here is some category and the vesicle types, which are lipoidal and non-lipoidal. In lipoidal, we can see here the liposomes, phaingosome, euphosomes, and novasome, oleosomes. They are standard lipid vesicle. There are some deformable and flexible. As I have mentioned earlier, that they have made some changes in existed liposomal structure. Okay, some, because of, with the help of some excipients, they make some changes like that. The circular structure and the lipid bilayer can get squeezed, can get deformed during the penetration in the skin, and after get penetrate into the skin, they get reform its size. Okay, so this is distinguishable character they have brought into this new vesicular system. So it can become a flexible as well deformable. There are some example like ethosome, transfersome, and so on. There are some herbal vesicular system as well, like phytosome, herbasome, and marinosome as well. And structural variants, we can see vasosome, cubosome, exosomes, specialized target for immunoliposomes, virosomes, genosomes, enzymosomes, as well as some protective stabilized like cryptosome, emulsosomes, lerosomes, leptosomes, carbohydrosomes, and others. In miscellaneous, some hemosomes, proteosomes, ketosomes, then ultrasomes, then proniosomes are also available for the in the literature, and the research work is going on so far. So their next vesicle type, that is a non-lipoidal, as we have mentioned earlier, that the lipid bilayer of this vesicle is not made up of phospholipids. Okay, instead of, they replace the phospholipid in niosome, they replace the phospholipids with the non-ionic surfactants. Okay, and here the cholesterol is help into the, it will help like a cement. I will explain in next slide, that is about the structure of niosome. Then aquasomes, bilosomes, and polymerosomes. Next part, that is the first very eminent, and we can say the first pioneer vesicular system in this novel drug delivery system, that is liposomes. The origin and the scope, it was first, liposome was first proposed by Alec Bangham in 1965. Liposomes are now clinically established diverse therapeutics ranging from anti-cancer drugs as well as biomolecules, basically due to its similarity with the cell. As we know, this is look like that it has a phospholipids bilayer that have resemblance with the cell membrane structure of our body, of our cell. Okay, inside that lipid bilayer, there is a presence of aqueous core, in which that aqueous core is formed by some hydration media. We will explain in the next part of this presentation. Okay, so the drug which is hydrophobic in nature, it will incorporate in the phospholipid part, that is hydrophobic. And with the drug which is a hydrophilic in a nature, that will absolutely uh, encapsulate inside the aqueous core, that is covered by the phospholipid layer. Okay, that it has liposome have a targeting capabilities, and the targeting capability that it can be this can be incorporated in an injection, and that is adaptable for surface, that is due to the adaptation to its surface modification, and achieve the precise drug delivery and drug targeting through IV route. So then there are some production challenges, some commercial successes hindered by high raw. There is a challenge about the raw material costing and complex manufacturing equipment, and the difficulties with sterilization come across while doing the formulation of the liposomes. Then the formulation limits. There are some formulation limit, that is a key performance that drawbacks the low encapsulation efficiency ability of liposome. There may be a chances of drug leakage due to some technical or due to some personal errors during the formulation of liposome. And sometimes the unpredictable release rate can be could have been occurred in previous research work. Then stability issues, liposome may have some stability related issues due to the poor stability and the rapid degradation in the gastrointestinal tract. There may be some chances of side effects, minor chances, not a major chances in that. The next vesicular system, that now the niosome, that is the restruct, we can call it as restructure, and we can call it we can call, they have made some changes in the structure of liposome that is presenting vascular system. The composition is completely that the non-ionic surfactants can form the bilayer structure of aqueous compartment. Okay, and it is also able to encapsulate hydrophilic as well as lipophilic drugs. Okay, and we can call it as an alternative for the liposomes. Okay, so the superior, it has some advantages over the liposome that is existing vesicular system. That the main advantage, it has a superior chemical stability as well as lower production cost, and it can have it has shown that the 84 months of shelf life period than the liposomes. Okay, the now we can say it has a cholesterol that is a very significant, it has a significant role because it acts as a membrane cement that increase the rigidity and control the permeability and prevent the drug leakage, which was the cons of the liposome vesicular system. Now that has been recovered in this niosomes. Okay, and it has a versatility to deliver anti-cancer drug, diabetes related drug, vaccines. It can be deliver the drug via oral, topical, as well as ocular route of administration. The commercial status in the market, the Dorzox, that is a drug particularly used for the treatment of glaucoma treatment, which is given by ocular route of administration, and the Lacome anti-aging cosmetic is also available in the form of niosomal preparations. Okay, now moving toward the next carrier, that is a transfersome. It is a, we can call it as a ultra deformable liposome as well. Okay, the main distinguishable characteristic or feature from the existing liposome is that in the, it is all completely similar to the liposome, but in transfersome, the distinguishing character played by the surfactant which are present in the lipid bilayer. These surfactants, these surfactants are responsible to squeeze the circular structure of the transfersome while entering into the very minute skin pores of the this dermis, this our skin layer. Okay, in that point, the surfactant played the role in which the surfactant helps to reduce its size, and it can reduce the size of the, it can reduce, or we can say the squeeze, it will squeeze this vesicle and enter into the skin layer, and then after entering into the skin layer, it will reach to the dermis layer, and after reaching at the dermis layer, it will get reformed in its original shape, that is a circular shape. Okay, so that's why we have called it here as an ultra deformable vesicle, and it has a good potential over the liposome, as it will reaches to the dermis layer, and it will have ability to deliver the drug, and to deliver the drug in a sustained and controlled release manner to the dermis layer, and through the dermis layer the drug will enter into the systemic circulation, and it has, so far it has a good rational over and perspective over the existing liposome. Okay, now the next vesicular system, that is a ethosome. Basically it is also similar like a liposome, as I already told that there are some variation, there are some, the scientists made some changes into this existing structure of liposome. Now in this structure it is completely similar to the liposome, but they have added some ethanol, as they, ethanol played a exceptional role in the composition of ethosome. Okay, so the ethanol has a great permeation enhancing ability. It will increase the membrane's flexibility, so it will allow to go deep, penetrate into the stratum corneum of the skin layer, and reaches to the dermis layer as well as the dermal layers of the skin. Okay, and it will have a therapeutic applications. It will use to dermal as well as transdermal application, and it can incorporate, we can incorporate both hydrophilic as well as hydrophobic drug moieties for the treating the conditions like alopecia, psoriasis, and dermatitis. And some marketed formulation are available of this ethosomal, for ethosomal preparations. There are like nanominox for the hair loss. In that, minoxidil is the main ingredient for the nanominox. Then supravir, then Nicilax, and the Silotype EF. Such marketed formulations are available in the market with this nanovesicular system, that is ethosomes, and binary ethosomes, that is we can call an advent variant combining ethanol with the propylene glycol. So when we add ethanol and propylene glycol during the formulation of this vesicular system, so it can give a binary ethosomes. So it will have superior ability of reaching skin, deeper skin layer, as well as some stability related points. Now the next vesicular system, that is a phytosomes. Unlike the liposome, the active herbal ingredient is a in this instead of phospholipid in with the phospholipids, the chemical constituent, that is a active herbal chemical constituent from the plants, it get attached with the phospholipids. Okay, it will attach with phospholipid and become the integral part of the vesicular membrane. That is a lipid bilayer membrane is present in this phytosome, and our herbal drug constituent linked with the phospholipid, and it will form the bilayer. Okay, so we can conclude that in this point that this herbal drug is remain in the phospholipid bilayer with linking with phospholipids. Okay, and it will enhances the bioavailability drastically and improve the intestinal absorption permeability of poorly soluble herbal drugs, example curcumin, silibene, etc. Then it also shows superior release as as it reaches to the site of action. It will show some enhanced release over the liposome or over the other vesicular system. Okay, then amplified efficacy. It will also boost pharmacological effect. For example, antioxidant, hepatoprotective activity while maintaining high safety profile. Now, therapeutic focus, it optimizes the pharmacokinetic profile of the body to target and treatment the hepatic, cardiac, as well as GIT related disorders. Okay, now next the part, that is some miscellaneous vesicular system on which the research work is still going on. There are some in some various literature you can find this all uh, vesicular system in which immense work, various famous scientists and researchers working on these vesicular systems for the goodwill of human beings. There are some example like archaeosome, cryptosomes with their compositions, euphosomes, unosome, virosome, bilosomes, and then leptosomes. Now, these are some again some miscellaneous examples of the zomal delivery systems: pharmacosome, vasosome, cytosome, then enzymosome, erythrosome, aquasome, polymersome, asparsomes, glycerosomes. And now the key component of this vesicular formulation, as we know the vesicular system, we are talking about vesicular system, we must know what are the key components, what are the excipients, what are the building blocks which block these cargo ships, which are helpful for the delivery of drug molecule and biologics to the deeper layer through the body for the betterment of the patient. So the component of the category, that is for example the structural building blocks for these vesicular formulations are the phospholipids, non-ionic surfactants in niosome, then phospholipids in overall vesicular systems, then block of copolymer in a polymerosomes. The function and their role, they will primarily basically the phospholipids is required for the vesicular membrane, that shell-like structure formation, and that will help to encapsulate the drug inside very well. Then some membrane stabilizer, as we have seen, cholesterol. The cholesterol helps in the niosome and act as a building block, as a cementing agent for there, and this will help definitely to increase the membrane rigidity as well as prevent the leakage of drug, early release of the drug, and enhances the physical stability. Some charge inducing agent, there are the example like DCP, that is a diacetyl phosphate, and sterlamine, that impart the surface charge, and this surface is very very essential during the characterization part of the vesicular system to calculate its zeta potential. Okay, then hydration media is a very very important point of this vesicular system. It will form the inside structure, that is the aqueous core, that is hydrophilic core, in which the drugs which are soluble in water, they will reside in that particular hydration media. That, and as we have mentioned, that it needs role, that it hydrates the lipid film and the aqueous core where hydrophilic drugs are trapped or encapsulated. Then there are some organic solvents: chloroform, ethanol, methanol. These are solvents are, I know these solvents are some show some toxicity, but the toxicity of these solvents can be reduced. The solvents can be completely removed with the help of GC, with the help of during the thin film hydration method. It get all evaporated, so there is no chances of presence of chloroform, ethanol, methanol. for that. And this solvent we are used to solubilize the lipids, surfactant as for the initial preparation of the phases, that is organic phase, and we remove afterward, as I have mentioned earlier. And then specialty additives like age activator, as I forget to tell you that in transferdome the surfactant which are helpful to squeeze or to deform this vesicular system, that is that can be called as age activator. And there is example of some age activator: sodium cholate, sodium deoxycholate, some span category surfactant, some twin category surfactants. Then the still agent, that is PEG, polyethylene glycol, cryoprotectant, that is a travelose. And their role is that they activate and provide the elasticity in transfer zone, that is sodium cholate age activator. PEG help to prolong the vesicular system, the blood circulation, so we will get the extended release of the drug moiety in the blood circulation. Then cryoprotectants, as we know, cryoprotectants are helpful during the freeze drying technique. Okay, now moving to the next part, that is the method of preparation for the vesicular system. Here are I have mentioned some method of preparation which are conventional preparation methods. First method is that the thin film hydration method. This method was proposed by Bangham in 1960, as he prepared the first liposome, that in that the organic solvent is evaporated with the help of evaporator, and after rotary evaporation the thin film is formed, and that thin film is get hydrated with the help of phosphate buffer saline solution, as we have mentioned in the particular previous slide. Then the reverse phase evaporation, in that the inverted micelles get formed in this reverse phase evaporation, and they collapse and then gel, and then the vesicle upon solvent removal, they with the help of we mix with the solvent, and the solvent is removed with the help of the rotary evaporator. And some key characteristics, that with the help of this method, that it has shows some high entrapment efficiency. Okay, now solvent injection method, basically the ethanol or ether in the lipid or organic phase of this uh, vesicular system. During the prior preparation, the ethanol get injected into this solution, so that the vesicle, or so that the solution get formed, and then the solution will be evaporated with the help of rotary evaporator. And with the help of this, the simple techniques, small unilamellar vesicles and residual solvent, use SUVs get formed, but the residual solvent removal is difficult, but this can be removed with the help of GC, and we can lower the toxicity risk. There is a detergent depletion method, and there is a dehydration and rehydration method. These are some conventional preparation method for the vesicular systems. Now there are some, here is the graphical, here the schematic representation how the reverse phase evaporation, there is organic solvent, aqueous solvent. After merging them, oil in water emulsion get formed. We have to sonicate this solution, then gel like matrix get formed, and we will with the help of rotary operation, we will get the solvent get evaporated, and we get thin film, as I mentioned earlier. And in that thin film get hydrated with the help of PBS. That PBS is also depend on the pHs according to the site of action where we wanted to deliver the drugs. So we will decide the PBS pH. Okay, this is the thin film hydration method graphic. This is a schematic representation, and this is a solvent injection method for the preparation of the vesicular system with the help of conventional or existed say, method of preparations. Okay, now the novel preparation methods are also there. There are some supercritical fluid technology, SCF, then dual asymmetric centrifugation method is there, membrane contractor technology, cross flow filtration, then freeze drying or double emulsion method, we can call it as. Then there is a microfluidic method as well as a curvature tune method. In supercritical fluid method, we use dense fluids like, for example, CO2 as a green solvent or anti-solvent to form a vesicle. It shows a high efficiency, okay, eco-friendly. It takes higher entrapment efficiency. It shows in that high stability of the particular vesicular system as well. Then in the DAC, that is dual asymmetric centrifugation, speed mix technology, that rotating the sample on the two axes that generates shear forces. And it show, and the key advantage of this method is that the homogenizes toxic sterile samples in a closed vesicle without any manual handling. So we can reduces the manual error on the human error during the preparation of the vesicular system. So then membrane contact technology, it help to in this the forces lipid, it will forces lipid and ethanol solution through pores into the flowing aqueous phase, so that the with the help of this, the key advantage we got uniformity, that instant and self assembly assembling structure, nanostructures of lipid vesicle get formed, and it will also reduce our time, and it is completely a time effective and cost effective as well. And cross flow filtration, that combines the detergent depletion with the tangential filtration. It removes the detergent rapidly compared to dialysis and enables the sterile and large scale production are some key advantages of this cross flow filtration. Freeze drying and double emulsion, the lyophilization of W by O by W multiple emulsion we have prepared early, and with that contain cryoprotectant. Okay, so in this method we got some key advantage, that is storage related, that is creates a stable powder form. So the powder form of this prepared vesicle will long lasting, and it has some stability related advantages, and it encapsulate the diverse drug, that is hydrophilic as well as lipophilic drugs. Then microfluidic method, that is very well known method for the preparation of the vesicular system. The nanoprecipitation via rapid mixing in a micro scale channels, that is called as chips, and it gives the major advantage, that is the precision, that one step process with the exact control over the size and reduce reagent cost. Then curvature tune method, that use the pH jump, that is the pH get jumps, that pH gets shift from 7.4 to 11 and 11 to 7 point come back to 7.4, that to induce the lipid curling. Okay, basically this the main advantage of this curvature tune method, that it is completely a solvent free method, rapid, simply simple, and avoid the organic solvent or thin film formation get done with the help of this method. Now these are the some novel preparation method. Now moving the next part, that is this is the schematic representation of novel vesicle preparation, that first A, this is the RESS, and these all methods as we have discussed earlier. Now, the drug loading techniques in the nanovesicle. As we all know that the nanovesicle can carry the biologics, drug molecules from towards the therapeutic, towards the site of action where we want its therapeutic effect. So we have some doubts, and now we can clear that doubts with the help of these novel techniques of the drug loading in the nanovesicle. There are some techniques like passive loading, in which it utilizes a concentration gradient for the drug incorporation into vesicle. It means the drug get entrapped, the drug get encapsulated during the phase of preparation, and then during in the organic phase before the it introduces to the thin film hydration method. Okay, in active loading, the drug is a drug in this loading, the energy driven mechanism is involved, so it will enhances the drug encapsulation efficiency as well. And then the remote loading, it usually utilizes the potential and pH gradient. Basically they are using gradients like potential gradient and pH gradient for the drug uptake inside the vesicular system. Okay, and there is next method as co-encapsulation, which means that at the same time, as we know, there is a two cores in the vesicular system, that is a bilayer. In the bilayer we can incorporate the hydrophobic drug because the nature of this lipid bilayer. Okay, and in aqueous core we can incorporate the drug which is hydrophilic in nature. So this is the main advantage of the drug loading technique, that we can co-encapsulate to we can co-encapsulate two drugs, so that the synergistic effect can be seen after entering this nanovesicle inside the body to the particular site of action. Now the next part, that is the release mechanism of therapeutic agent. We all are a little bit excited how the drug is released and how the drug reaches to the site of action. There are many steps, but I have concluded that many steps into the three steps. That is step one, that the nanovesicle encapsulate the drug and release the drug upon reaching to the target site. As we have mentioned, whether it is ocular route, whether it is IV route, whether it is a topical or transdermal route, the vesicle will come in contact with our skin layer. Okay, after come in contact with the skin layer, okay, then it will enter into the skin, or it will enter into the eye or into the blood vessel, but the drug will not release, early release will not seen. In this case, the drug will the vesicular system will reach to the perfect site where the targeted site or infected tissue site. At that site, the drug will the capsule will reach, and at that point it will release its drug molecule or the biologic. Okay, and the next step two, that is external. Sometimes the external stimuli such as pH changes, temperature changes will also release the encapsulated therapeutic agents at the site of action. at the particular site where therapeutic action is needed. And in step 3, the targeted delivery allows us for the localized action. We have already mentioned that it will allow completely only localized action at the particular infected site, and it won't harm the healthy tissue, healthy cells of the body. Okay, at it, and that's why it will minimize its side effect and enhances the targeted treatment efficacy of the drug delivery system. Then the next part, that is the physicochemical characterization. We have prepared the as from this presentation, we have till now we have seen what are the structural building blocks of the vesicular system, what are they, how they release. Okay, now we have to I have to throw some light on the physicochemical characterization of this vesicular system. Okay, after formation or after formulating this vesicular system, there are important characterization related aspects, such as there are some parameters like vesicle size and distribution. Okay, the vesicle size, basically we know the vesicles, this nanovesicular system are completely in the ranges of nanometers. Okay, like 330 20 to 100 or in nanometer, in this range. Okay, so for this the vesicle size is determined with the help of dynamic light scattering, that is a DLS. Okay, then microscopy, we can look its structure with the help of microscope, that is TEM and AFM, that is TEM transmission emission microscopy. Then atomic force microscopy, as well as analytical ultracentrifugation AOC technique is also there for the vesicle size and distribution. Then some key insight of that, it will determine the average diameter and the polydispersibility index of the vesicle. Okay, so for the drug that is required for the drug encapsulation volume and in vivo clearance. Okay, then morphology of the particular vesicular system can be done with the help of cryo-TEM. Okay, freeze fracture TEM, then AFM, that in a hydrated state. Okay, it will the key insight of this particular characterization is that it will visualize its shape, that is a spherical, tubular and lamellar, as well as without staining the artifacts. Okay, then surface charge, the zeta potential, as I mentioned earlier, that the zeta potential, that is surface charge, is very very important. That is electrophoretic light scattering, or we can in the DLS instrument there is a they have made a provision about the ELS, that is electron light scattering. With the help of that, we can determine this their zeta potential. Okay, it measures the potential and shear plane and high value, that is greater than plus minus 30 millivolt. Okay, it will help to determine the stability of the particular vesicular system in this in its form. Okay, then supramolecular organization with the help of SAC study and SAN studies. Okay, small angle X-ray scattering and the small angle neutron scattering. With the help of this technique, we can analyze the bilayer thickness and the lipid packing, as well as the fluid gel state of the membrane. Okay, now the next parameter of the characterization, that is the encapsulation efficiency, we can determine with the help of separation, that is dialysis and ultracentrifugation. And analysis is done basically on the instrument like HPLC, UV visible and fluorescence spectrometer, as well as the purpose and key insights of that. It will quantify the percent drug interact inside the vesicle and the free drug which is present outside the vesicle. So we get a real crystal clear idea about the how much concentration of drug is present inside the vesicular system, and it is critical to assessing the formulation efficacy as well. Then we can determine the lipid content with the help of Barlett assay, that is phosphorus with the phosphorus, and HPLC ELCSD assay for the lipid content, that quantifies the total phospholipid concentration to account for the losses during the preparations. Okay, in vitro drug release with the help of dialysis bag method, as well as continuous flow USP type 4 model, and it measures the rate of the drug release over the time in controlled buffer environment as well. Then the stability related issue, physical and chemical, size zeta monitoring over time, chemical assays, hydrolysis, oxidations are done. Okay, it will assess the shelf life, aggregation, drug leakage, and the lipid degradation, oxidation, hydrolysis point related to our vesicular system. Okay, the in vitro performance assay for the nanovesicle, there are some in vitro assay, that is a drug release studies. Drug release study we can done with the help of France diffusion and USP apparatus 4 model. It will help to understand the release kinetics of this drug molecule through the nanovesicular system. Then cellular uptake, we can determine how effectively the vesicle enter the target cell and identify their intracellular pathway. If this can be done with the help of fluorescence microscopy, then confocal lens microscopy. Quantification can be done with the flow cytometry, FACSS. Then cytotoxicity, they assesses the biocompatibility safety of the healthy cell and killing the efficacy on cancerous cell for the cancer treatment by the nanovesicular system. For that we can do the MTT assay, that metabolic activity alarm blue, then live dead staining with the help of nanovesicular system. Then barrier models that stimulate the biological membrane to predict the transport of drug through the vesicular system across the gut and skin or blood brain barrier. Okay, and for that we have some common method, that is get Kakutum cell monolayers. Okay, then BBB, that is a transwell co-culture system, then general PAMP, a artificial membrane, then serum stability we can done. Before that method we use incubation of FDS, human plasma at 37 degrees Celsius, and we can monitor the side chain DLS or drug leakage. Okay, now the next part, that is the pharmacokinetic and biodistribution. Nanovesicle basically enhances the targeted drug delivery, as we have mentioned, and we are talking about it so far. That leading to the increased drug localization to the desired site. As we know, we don't want to harm the healthy cells. We want to treat the unhealthy cells and to treat the cells which are damaged due to this disease particular progression. Okay, so in that particular desired site, we can localize the drug and we can target its action. So this can alteration of pharmacokinetic and biodistribution of drug with the help of a nanovesicular system. Then their size and surface modification is enabled to control release profiles, as well as improve the therapeutic outcomes and reduce the potential side effects in the body that have been seen in the conventional drug delivery systems. Okay, now the safety and immunogenicity and toxicity. There are some toxicities related to vesicular system. This is pseudo allergy, we can call this as CARPA, that is activation of the complement system innate immunity immediately upon injection. In they have some cases that the injection of a ambiosome, that is very well known example, okay, of the liposome, they have seen some activation of innate immunity immediately. Okay, but this impact is a very acute hypersensitivity. They have some shown some flushing and hypertension in that. Then anti-PEG antibodies. Okay, next challenge, that is the body produces the IgM or Ig antibody, as we know, for the innate immunity system in our body. They have, as we have mentioned, the PEG coated some vesicular system are present. Okay, so if these vesicular system when enter into the body, sometimes the body produces antibody against it. Okay, the impact that ABC phenomenon, that accelerated blood clearance may got get to with the patients. Okay, where subsequent doses are cleared and rapidly by the liver and reduces the efficacy of the drug. Okay, then if excipient sometimes some excipient show some toxicity, but that can be minimized with the help of completely removal of this residual solvent or surfactant, and that will help to to increase its efficacy into the patients. Then the next part, is regulatory consideration. The preparation and the formulation of a nanovesicular system basically adhere to the FDA regulation. Okay, it ensures the consistent manufacturing as well as the standards of all the nanovesicular batches, and it will conduct the intensive toxicity studies of a nanovesicle on the human in the human trial to minimize the or to reduce the risk after getting after the treatment in the patients. Okay, the proper documentation should be maintained for the validation as a validation process for the formulation related documents of this vesicular system. Then efficacy, that demonstrate the target delivery efficacy through the clinical study results. Okay, now the next part, that is the marketed formulation. Here are I have taken some cutouts from the articles where the very well-known and well-known and famous examples of marketed formulation, nano formulation. These nanovesicular containing formulations are present in the market. The very well-known example is that the antifungal drug, that amphotericin B, its injection, that is ambisome, is present in the market. In that, the amphotericin B is encapsulated in the liposome, and it shows a distinguished treatment in the distinguished role in the treatment of this fungal infection. There is some anti-cancer drug injection, that is a doxil, as we have seen in this PPT, that is a doxorubicin hydrochloride liposome injection is present in the market for the treatment of this cancer disease. Then some nanovesicular formulation under clinical. Here are some cutouts from the articles where are various like nanosomal formulation as well as some liposomal formulation as well as some ethosomal formulation which are under the clinical trial, and after clearing their clinical trial data and clinical trial regulation, they will also come in market, and it will also help to the human welfare for the treatment of various diseases. Okay, now some challenges in nanovesicular system we have mentioned earlier, that stability related challenges, they can be overcome with the help of some novel techniques. Then bioavailability, sometimes the sufficient drug release may not be occur, but that we can also improve with the help of some technique and improvement in this particular bioavailability related risk. Then scalability, regulatory, targeting, and cost. Cost for in the point of view of cost, there is a high production and processing cost for that can be limited and widespread adoption of this technology. Then targeting, sometimes we need to very precise for deliver the drug at the actual site of action for to the body to remains in a hurdle. Okay, now the next part, that is the cutting edge research, and we can call it what are the future directions. Okay, now in the innovation area, we can gone through these images which we have pasted here in the presentation. Okay, there are some theranostic nanovesicle which I have mentioned as earlier. The theranostic means that will help to give the therapy, that is treatment, as well as it will diagnose the particular disease, and it will release the drug according to and we can see this real time imaging, and we can conclude the how the drug is released and how much amount of drug got released in that particular site of action. Then innovation area is stimuli responsive vesicles, that is we can call it smart vesicles. Okay, the release cargo only upon the specific triggers. Okay, acidic, tumor, peers, ultrasound, these are the stimuli responsive. We can prepare the vesicles. Okay, the goal that on demand delivery, it will help to prevent the premature leakage and ensure the drug release only at the target site. Okay, then biomimetic and the hybrid systems. Okay, camouflaging vesicles with the natural cell membrane, that is RBC, platelet, or exosomes hybrid can be present. Okay, now stealth mode, that is a trick the immune system to avoid the clearance, enhances tissue specific homing. Okay, as we have seen that there are some challenge that the some antibodies get from inside the body against this vesicular system, but we can also do some new changes in the particular vesicular system, so that can be avoided and the treatment can be given with the help of vesicular system. Then gene editing, mRNA tech delivery. Okay, next gen lipid, that is a nanoparticle, that is liquid nanoparticles. Okay, optimized for the CRISPR Cas9 and mRNA payloads. Okay, genetic therapy enables safe and non-viral editing, genetic defect, and rapid vaccine development. Theranostic, as I mentioned earlier, the co-loading vesicles with both diagnostic agent, that is quantum dots, and the therapeutic drug that will help to cure from that particular disease or particular infection. See and treat. Okay, it allow, that is a goal and impact of the theranostic, that allow the real-time monitoring of the tumor in the cancer treatment and its regression and the drug accumulation simultaneously at the point of tumor in a cancer treatment. Okay, then brain targeting, that is targeting of a BBB, that is triple B, service modification with the help of ligand, that is transferrin glucose, that will help to cross the blood brain barrier, so that the diseases or the infection related to the CNS will get cured with the help of this nanovesicular drug delivery system as a personal nanomedicine. Okay, for the treatment of very famous and very curable uncurable disorders like Alzheimer, Parkinson's, and glycoblastoma. With the help of theranostic nanovesicles and personal nanovesical medicine, we can get a success to treat these deadly diseases. Okay, now the concluding remarks and the takeaways from this today's talk about the nanovesicular drug delivery system, that it is an innovative drug delivery system, as we have discussed earlier. Now nanovesicle, because it enhances the precision and the efficiency of the targeted drug delivery, site specific target drug delivery. Okay, without harming the healthy cells. Okay, now versatile application, it can carry both as well as hydrophilic as well as lipophilic drug moieties inside it, and ongoing research is crucial to optimize the nanovesicle formulation for the clinical application. Future market and growth, the market of nanovesicle drug delivery system based therapies is expected to grow significantly in coming years. So we need to collaborate with the various pharmaceutical companies, that industry and academia collaboration should be there for the real world application, and initiate the trial to test the safety efficacy in human subjects, and need to pursue the grants targeting advanced drug delivery systems. Okay, and that's all from my side. Thank you so much. I'm really happy and really glad to all the authorities related to this webinar, organizing committee, Mr. Urmesh Bhamre sir, the coordinator of today's webinar, IQAC coordinator, Honorable Vice Principal Sir, Dr. Pingle Sir, as well as our beloved Principal, Dr. S.V. Amrutkar Sir, and all mentors present over my alma mater, my all faculty and non-faculty, non-teaching staff. Thank you so much for providing me this opportunity to come in front of you in my alma mater to deliver this talk and as a spokesperson or speaker for today's webinar. Thank you so much, ma'am. That's all from my side. If there are any question, you are free to ask.
Yes, Saurav, can you see the question on a screen? In the chat
Yes,
box?
yes, sir.
Yes.
Can this
Can
technique replaced by AI technology or? Yes, Sanjana, I got your question that these techniques you are talking about technique of formulation, na? I think so. Yes, yes. I think she is talking about the formulation technique. It can be, but the research work is going on in this manner to replace these techniques. Already they have replaced these conventional techniques, but now they have to replace these technologies with the help of AI techniques. But in that point, we have to look its drug efficiency capacity. Its basic, can we get that particular size or this aspect? We need to look for this. Look after that they're implementing this AI technology. So it can be possible. Yes, the answer is yes.
okay. Check करा दिसते. Okay, thank you, Saurav, for delivering an insightful and well-structured session on vesicular drug delivery systems. Your explanation on
Thank you, ma'am.
liposomes and advanced vesicular carriers have greatly enhanced the understanding of students. We appreciate the clarity and real-world examples. Your session has inspired the students and researchers to explore the further research and innovation in drug delivery technologies. On behalf of organizing committee, principal and faculty members of MSG Copper, I am very thankful to Mr. Sourav Suryawanshi for his talk on nanovesicular drug delivery systems, a pioneering the future of precision therapeutics. Uh, Saurav, kindly accept the certificate of appreciation.
Thank you, ma'am.
Yes, thank you very much.
Thank you so much, ma'am. Have a good day, ma'am.
Yes, yes. With this, I conclude the second session is over.